We're going over today how we isolated our RNA samples using the Quitogen kit. We first took out our UV and non-UV samples out of the refrigerator or out of the freezer and allowed them to thaw until they became a liquid again, and you could no longer visualize the pellet. In the first step of the kit, we added the buffer RLT, which is the lysis buffer, to each of our samples, and then we used a 20 gauge needle and a syringe to basically mix the lysate. We took 350 microliters of each 5 times up and down with the needle to pull the liquid through. Then we disposed of the needle and added 70% ethanol to each of our tubes and centrifuged those and discarded of the flow. And then we added buffer RW1, which is one of our wash buffers, to each of our tubes and centrifuged that and discarded of the flow. And lastly, in the kit, we added buffer RPE, which is another wash buffer, and we centrifuged that, discarded the flow, and then we centrifuged again and discarded of the flow. And then we added water, RNase-free water to the column, to our centrifuge tubes, and centrifuged that again. And we were left with around 28 microliters of RNA in each of the tubes, which will be used to find the concentration used in the nano drop. That's it.